Hello students, I am Akshay Deshpande and welcome to another biology class. The concept of metabolism. Okay, so now if you consider any living organism, okay, any living individual, you can see that there are numerous organic molecules. And we know that any molecule that is derived, okay, if it is derived from it is derived from carbon, then we call it as what? Organic molecule. We all know that, right? So there will be what we can say n number of molecules, numerous molecules occurring at a given moment of time in the case of living organisms. Okay, and we also see that these molecules are present at one particular concentration. Okay, it is said that it is a stable concentration. It is a stable concentration, right? So we can say that none of the molecules are excess or low. Okay, none of the molecules are excess or none of the molecules are in a low concentration, right? They are always maintained in one particular concentration, in a stable concentration, we can say, right? And generally we can actually measure this concentration in a given unit called as the mole per cell. Okay, it is called as the moles per cell, or we can even say it as the moles per liter. Okay, it is moles per liter or moles per cell. That is what the units that we mainly use to describe the amount of the organic molecules present in one particular concentrations, right? So now we observe that these molecules are actually having a turnover. Okay, we are having a concept called as what? Metabolic turnover. Okay, the term metabolic turnover means that in a cell, we observe that the biomolecules are constantly changed to some other biomolecules, right? Yes. So these reactions, okay, we call them as what? Turnover kind of reactions. Okay, so it does not mean that, for example, if you consider amino acids, right? If you consider amino acid, there is no rule that this molecule inside the cell will remain as it is. Okay, it will remain as it is. No. Okay, it will never remain as it is. Okay, it means that if an amino acid is present inside the cell, it is not remaining as amino acid only, right? It will be used for mainly formation of protein. Okay, it is mainly used for formation of protein, right? Yes, it may be used for the formation of the protein. Or similarly, when the protein is actually formed inside the cell, there is no rule that the molecule will be present in the form of protein only. The protein may be broken down to give us the amino acids back. It may break down to give us back the amino acids, right? So we can say that amino acids may be joined to form the proteins, and proteins may be broken down to give us the amino acids. So there is none of the biomolecules will remain in a same state. Okay, they will be constantly changed into from one form to another form, and this is what we mean by the term turnover, right? We have mentioned it over here. Constantly the molecules are being changed into from one form to the another. right? So now we can see that these making and breaking kind of reactions are constantly occurring, right? See, for example, we have discussed about the amino acid, right? I will take one amino acid and I will join it with another amino acid, right? And I will add one more amino acid and I will add one more amino acid. So don't you think that these kind of reactions are actually called as making reactions, right? They are actually called as the making reactions. These are actually called as the making a kind of reactions, or we can even say that they are actually constructive reactions. Yes, so these kind of reactions are actually the constructive kind of reactions. Now there are some reactions, we also call them as breaking the reactions. Exactly, right? If I break the protein, if I break this particular bond, if I break this particular bond, and if I similarly break this particular bond, what is the result? I can see that I will get independently amino acids. I will get independently amino acids, right? So I will get free molecules of free randomly formation of amino acids, right? So breaking down of a protein to release the amino acids, we call them as the breaking breaking reactions. We call them as breaking kind of reactions, right? So collectively we can say that the making kind of reactions, making kind of reactions, and breaking kind of reactions, we call them as the sum of these two kind of reactions are called as the metabolism. It is called as the metabolism, right? If I use the term making kind of reactions and breaking kind of reactions, the sum of these two reactions are called as the metabolism, right? So here that is what they have mentioned over here. That is the metabolism is a sum of two types of reactions. The first one, it is called as the metabolic reactions, and another one is called as the anabolic reactions, right? So here anabolic reactions are mainly used for making a kind of reactions, right? So anabolism, right? Any kind of reactions where two molecules are joined together to form more complex kind of molecules, right? If I fuse or if I attach two simple kind of molecules, that results in the formation of more complex molecules, right? So we can say that the anabolism results in the formation of complex molecules, complex molecules. Then it is called as mainly the anabolism, right? And similarly I will write one more that is called as the catabolism. Catabolism, right? So in these kind of reactions, what actually happens, you can see that they are called as the breaking kind of reactions, which result in the formation of the simple molecules, right? It results in the formation of simple molecules, right? So if there is a formation of complex molecules, then we call it as anabolism. If it is resulting in the formation of simple molecules, it is called as the catabolism, right? So hence we can say that the sum of these two reactions, that is the catabolic reactions and the anabolic reactions, we call them as the metabolism. The sum of these two reactions are called as the metabolism, right? Of course we will try to understand about catabolism and metabolism, that is anabolism and catabolism in shortly also, right? So these kind of reactions, it is called as the transformation reactions. We are having some examples. If I remove the if I remove the carbon dioxide from the amino acid, you can see that an amine is actually formed. If I remove the carbon dioxide from the amino acid, the result we get is actually the amine, right? Similarly, breaking of what we can say glycosidic bond. The breaking of glycosidic bond is actually what results in the formation or release of water, right? So it's a dehydration kind of reaction, right? So hydrolysis of what we can say glycosidic bond. We all know where do you find the glycosidic bond. The glycosidic bond are mainly formed in the case of the carbohydrates, that is the polysaccharides, right? In the case of polysaccharides, you mainly see that it is mainly having what kind of bonds. that is the glycosidic bond. Okay, an example for polysaccharide we are having is the cellulose, right? We have studied about cellulose a lot. I will take one glucose molecule, attach it with another glucose molecule, and similarly with another glucose molecule, and so on. So this bond, what we are looking over here, are actually called as the glycosidic bond. And if I break this bond, then it is hydrolysis of glycosidic bond, right? So if you are removing, okay, removing or attaching, right? So this is actually converting one type of molecule into another kind of molecule. Hence, such kind of reactions are called as the transformation. Transformation, what is the term? Transformation mainly means, it means that we are converting it to the another form. Okay, hence they are called as the transformation reactions. Okay, so one example we have discussed over here is what? Mainly removal of certain groups. For example, if I remove the carbon dioxide, we get is the amine, right? So if I break the glycosidic bond, we get simple sugar molecules, that is the glucose, if we consider cellulose as the example, right? Yes. Now we can observe that metabolic reactions can never occur in isolation, right? They never occur alone. Okay, they are always a series of linked reactions. Interestingly, all of these reactions are actually linked reactions, right? See, we can consider one of the well-known and well-studied examples from our classes, current class. For example, we can look into the energy metabolism, okay? Energy metabolism, yes. Because you people will study about these examples a lot in upcoming sections, okay? See, we can consider about the glucose, right? Yes. We can consider the glucose. Glucose is actually broken down into a series of 10-step reactions. Okay, it is broken down through a 10-step reactions. And what is the final product we get? We get a pyruvic acid. Okay, we get pyruvic acid as our final product. Okay, now interestingly, pyruvic acid, which is the last product after breaking down the glucose, here it does not stop. Okay, so again, the fate of the pyruvic acid depends upon whether there is a presence of oxygen or whether there is no oxygen, right? So if there is a complete presence of oxygen, what happens? The pyruvic acid actually enters into the next stage of a series of reactions called as the TCA cycle, right? Or also called as the Krebs cycle. Yes, we have studied about this cycle, right? Yes, you will study about mainly this in the respiration. That is the tricarboxylic acid cycle, also called as the Krebs cycle. Right? Yes. So we can say that the pyruvic acid is a molecule that is actually involved in linking one type of metabolic reaction with the next type of metabolic reaction, right? It is a linking molecule, right? It does not stop only, the glycolysis does not stop just after the formation of the pyruvic acid, right? The actual process of breaking down the glucose into pyruvic acid is actually stopped, right? But however, you can see that the pyruvic acid, after it is formed, the fate of this pyruvic acid ultimately depends upon whether there is presence of oxygen or not, absence of oxygen. And if there is oxygen, then it will enter into the next cycle, that is the TCA cycle, right? And finally, the products of the TCA cycle and the glycolysis, we get mainly some of the molecule called as the NAD, and one more we are having is the FAD. Okay, it is mainly NADH2 and FADH2, right? So these molecules will finally enter into the last stage of respiration. It is called as the electron transport system, right? So we can say that this is the last phase of the aerobic respiration, right? So you can see that these set of reactions are actually linked reactions, okay? And we generally call these linked reactions as a metabolic pathways, okay? It is what? Metabolic pathways. So generally these metabolic reactions or metabolic pathways are either may be linear, or they may be circular, or they may be again what? They may be interconnected, right? So we have discussed about the pyruvic acid, which is being involved in the TCA cycle, and ultimately it is the product of the glycolysis, okay? So we can say that the next stage of aerobic respiration is linked by the pyruvic acid. Hence it is a true statement for our discussion, okay? And also we said that the reactions may be circular, circular, or they may be even linear, right? So what do you mean by linear kind of reactions? For example, A is converted into B, B is converted into C, C is converted into what? D, right? So these kind of reactions are called as the linear reactions. Exactly. Right? But whereas some of the reactions may also be cyclic or circular. Okay, circular in the sense they are mainly referring to the term cyclic in nature. Okay, so it means that, for example, A will give us the B, B will give us a C, C will give us a D, and D will again come back and result in the formation of what? A molecule, right? So these kind of reactions, we call them as what? A circular or a cyclic, what we can say, type of metabolic reactions. Okay, so there is no specific rule that the reactions should be occurring either only linear or either only circular. Okay, and they can be either linear or they can be either circular, and similarly many of these pathways are interconnected like traffic junctions. Okay, yes, they are connected as traffic junctions, right? See, for example, if there is a road over here, consider this is a road, right? Yes. So here we are having around 5 road, and at the beginning of each road we can see that there is a traffic signal over here, right? So the switching on and off of the lights which are present at each road will control the moving of traffic from this road to this to this, and this to this, right? Simultaneously vice versa, from one particular road to any other particular kind of road, right? So we can say that these are traffic junctions. If they are interconnected, they are called as the traffic junctions. And interestingly, there is no jam occurring inside these traffic junctions. Okay, no jam, right? None of the molecule gets struck or stopped in between during these processes, metabolic reactions. Okay, they are highly coordinated and work in a very beautiful fashion. Yes, right. So now this is what we call as the flow of metabolites, right? So let us consider the glucose reaction only. Okay, for example, if we consider first few steps, so glucose to glucose 6-phosphate, glucose 6-phosphate, right? Yes, from glucose 6-phosphate we get the fructose 6-phosphate, right? Yes. So this is what we call it as the flow of metabolites, right? It is always converted into one molecule to the another molecule, which is called as the turnover. Okay, and finally after the product formation of one metabolic reaction, that is the pyruvic acid, it immediately enters into the next stage of cycle, that is the TCA cycle, right? So movement of we can say movement of the pyruvic acid into the TCA cycle or the tricarboxylic acid cycle is called as the flow of metabolites, right? And this state of continuously moving of metabolites is called as the dynamic state of body constituents. They're called as the dynamic state of the body constituents. Dynamic means what? Always constantly moving. Okay, not stationary, not stagnant. That is what the term, it is exactly opposite of the dynamic. Okay, stationary or stagnant means that stopping. Okay, dynamic means constantly moving, right? This is the meaning of dynamic. Okay, dynamic state of body constituents. It means that ultimately what are the constituents of the cell or living organism? Biomolecules, right? So these biomolecules will be constantly moving into one form into the another form, or they may be moving into what? They may be moving into the from one type of metabolic reaction into another type of metabolic reactions, right? Now we also see that almost all kind of organic reactions, okay, metabolic reactions, what you see, the reactions occurring inside the cell, all of them are actually catalyzed reaction. Okay, all of them are what? Catalyzed reaction. Okay, so you will study about the catalysis more detail in the last class of the biomolecules, where you will study about the enzymes more properly, right? So here in the case of a catalyzed reaction, you require a molecule called as the catalyst. Okay, you require a molecule called as what? Catalyst. Okay, so what does a catalyst do? Catalyst basically alters the rate of reaction, right? it mainly alters the rate of reaction. So what do you mean by the rate of reaction? It means that either the reaction is speeded, okay, either it is speeded up, okay, either it increases the rate of reaction, or either it may be slowed down, okay, either it may be what? Slowed down, right? So this is called as alteration of the rate of reaction, okay, and any molecule that is involved in altering the rate of reaction is called as an catalyst, right? Yes. So as they have mentioned over here, metabolic reactions are catalyzed reactions, and catalysts alter mainly the rate of reactions, okay, and some proteins are actually having catalytic power, hence they are referred as enzymes, right? So we will be studying about enzymes in the last class of the biomolecules, right? That is the next class. We will be mainly studying about the biomolecules, right? So in the cell, okay, in the cell, almost all the enzymes that we know, major of the enzymes that we know are none other than the proteins, okay, but it doesn't mean that all of the proteins are actually enzymes, right? We have discussed this. For example, collagen is actually a protein, but it doesn't make it as an enzyme, right? But however, rubisco, catalase, peroxidase, all of these are actually enzymes, and they are also proteinaceous in nature. They're also what? Proteinaceous in nature. We are having one exception to this particular rule, that is called as the RNA. Okay, mainly the RNA. Okay, there is an enzyme that is derived from the RNA molecule, and it is called as the ribozymes. It is called as the ribozymes. Okay, let me write it more clearly over here. It is called as the ribozymes. Okay, now ribozymes are actually exception to the concept of what we can say enzymes, okay, because we can say that almost all of the enzymes that we have recorded in the nature are mainly proteinaceous in nature, okay, they're mainly made up of proteins. But however, ribozymes are not made up of proteins, okay, at least the part that is involved in the catalysis, okay. What do you mean by catalysis? See, remember, in the case of enzymes, okay, any enzyme, if you consider any enzyme, okay, this enzyme is having the catalytic property. It is having the catalytic property. And what do you mean by catalytic property? It means that formation of bond or breaking of bond. Okay, either you're going to break the bond or either you're going to form the bond, right? So this property is called as a catalytic property, right? So ribosomes have, that is the ribozymes, okay, ribosomes are none other than the ribozymes only, okay. So the ribozymes are actually having this catalytic property, okay. So we can say that the part of the ribozyme which is having this property, that is the catalytic property, is not made up of protein. It is completely RNA in nature, okay. So it is said that ribozymes actually have proteins, okay. They have protein molecules, okay. They actually have the protein molecules. But interestingly, as I said, though ribozyme has some protein content, but this protein that is present in the ribozyme is not having catalytic property. Okay, it has been proved. It has been experimentally proved that ribozymes, mainly the RNA part of the ribozymes, are involved in the catalysis. Okay, so any example for ribozyme? Yes, we are having, that is the ribosome. Okay, ribosome is an example for what? A ribozyme. Okay, I can understand it is quite congested. Okay, if I erase, the whole thing can go. Okay, so what we can do is we can just erase some part over here. Yeah. Okay, so let me write the same point again over here, that an exception to enzyme, that is a proteinaceous enzyme, is the ribozymes, right? Ribozymes are mainly made up of RNA molecules, and these ribozymes have the catalytic property. Yes, it is mainly having catalytic property, and an example for ribozyme is the ribosome. Remember this, okay? Some questions have appeared in the previous question papers regarding asking which of the following is not made up of protein and yet is having a catalytic property. Okay, it is mainly ribosome. Okay, so the option they may give you as a ribozyme or a ribosome, all of it actually means the same.